Mouse Tumor Biology Database, MTB, is a comprehensive, expertly curated database of mouse models of human cancer and patient-derived xenograft PDX cancer models. MTB follows the standard nomenclature for mouse strains and genes in compliance with the International Committee on Standardized Genetic Nomenclature for Mice. MTB contains detailed information from the published literature and direct submission on tumor genetics, biology, pathology, clinical responses, and enables integrated complex queries of these data. In this video, we will demonstrate several powerful query tools available in MTB. We will show how to use the mouse model summary table and the faceted tumor search tool. We will demonstrate how to use the PDX model search form. Lastly, we will illustrate the functions and utility of the dynamic tumor frequency grid. The mouse model summary table provides counts of mouse models in MTB for the types of cancer that have the highest reported mortality in humans according to the American Cancer Society. Model counts are restricted to reports where sample size is 20 or greater and tumor frequency is 80% or greater. Clicking on a link in the model summary table opens a faceted search summary results page for the mouse models for a type of cancer, for example, lung cancer. Here is a faceted search page that lists all lung mouse tumor models. The search results can be refined by selecting parameters from the faceted list of annotations, for example, adenocarcinoma. Results are reduced from 44 to 14. Our search can be refined further by selecting additional parameters. Here we request only those mouse models for lung adenocarcinoma that harbor germline mutations in the KRAS oncogene. We can broaden the search results by deselecting any of the previously selected filters. Here we deselect the lung cancer constraint, which results in a listing of all adenocarcinoma mouse models in MTB with mutations in KRAS. Each row on the results page has a link to the reference describing the model and to a model summary report. The model summary report includes details about the mouse models, such as genetic background, observations of metastases, and other useful information. Next, I will illustrate how to search for PDX models and associated data. Information on patient-derived xenograph models can be accessed from the mouse model summary table or from the PDX search page. Here we will navigate to a list of lung PDX models by clicking on the link in the mouse model summary table. The search results displays a summary of all of the relevant lung cancer PDX models in the database. We can change the search by selecting the Search Again button. Selecting lung in the primary site pick list, adenocarcinoma from the diagnosis pick list, and entering KRAS into the gene search window will return all adenocarcinoma PDX models with KRAS mutations. The PDX search results page lists basic model information as well as a summary of data types available for a particular model. Clicking on a model ID will display detailed information about a PDX model. The PDX model detail page lists information about the patient characteristics and the type of PDX model generated, as well as a summary of other types of data, including genomic data for the engrafted tumor, histology images, results of dosing studies. Finally, I will illustrate MTB's tumor frequency grid. The tumor frequency grid can be accessed from the menu bar. The tumor frequency grid summarizes the inherent cancer susceptibility of different inbred strains of mice as a heat map. The color of the cell indicates the reported frequency of tumors in a particular organ or tissue. The darker the color, the more frequent the tumor type in a particular mouse strain. Details can be accessed by clicking on a specific cell. Tumor search results summarizes the frequency data for a strain by organ combination represented in the grid. Additional data for each model can be found by following the summary link. When available, annotated images of the tumor pathology are provided. MTB also has additional search options and resources not illustrated in this video, including advanced search forms, searching for mouse models using human gene symbols, interactive mouse cancer QTL maps, indexing of publicly archived gene expression datasets for mouse models of human cancer, an immunohistochemistry resource page, whole slide scans of lymphomas and specific staining patterns, and a pathology image submission page. In summary, the Mouse Tumor Biology Database provides access to detailed data on mouse and PDX cancer models and enables customized searches of these data. Examples of the kinds of questions that can be asked include: what strains of mice develop lung adenocarcinomas at high frequency? What types of endogenous mouse tumors harbor germline KRAS mutations? What treatment response data are available in lung PDX models? The MTB is supported with funding from the National Cancer Institute. The MTB can be accessed at tumor.informatics.jax.org. Send questions and comments to user support at the following URL.